Hello everyone. In this video we are going through the topic of insulin. So insulin basically synthesized and released from the pancreas. So insulin basically released from the beta cells of the isolates of Langerhans. Other type of cells also release some other substances. For example, alpha cell release the glucagon and delta cell release the somatostatin. So these three hormones released from the different cells of isolate of Langerhans. So structure of the insulin, it's a polypeptide hormone. The structure of the insulin is similar with other species insulin, for example beef and pork, which can be synthesized by the DNA recombinant technology. The detailed structure of the insulin you are going to learn in the biochemistry part. So we are just going through the physiology of the insulin hormone. So how the insulin synthesis takes place. As you know that insulin is a polypeptide hormone, so the same mechanism of the protein synthesis works over here. So insulin synthesis takes place inside the rough endoplasmic reticulum as a pre-proinsulin. Later it is converted to proinsulin, and in the Golgi apparatus when it is packaged in the form of insulin. These are packaged in the granule-like structures or pouches of the insulin. You already know that circulates in unbound form. Half-life is 6 minutes, so rapid removal. And degradation, liver is the main site of the degradation of the insulin and other tissues. For example, kidney. It's a relatively less amount of the insulin is degraded by other tissues, but the main degradation takes place in the liver. Now mechanism of insulin secretion by the beta cells. This one you are seeing, it is the beta cell. Once the glucose concentration increase inside our body, for example after meal condition, this glucose enter inside the beta cell of the isolate of Langerhans via the GLUT2 receptor. So the concentration of glucose is increased inside the beta cell, and this glucose is converted to glucose 6-phosphate via the enzyme glucokinase, and with the process of oxidation this glucose 6-phosphate forms a number of ATP. So the level of ATP becomes increased inside the beta cell, and this blocks the ATP sensitive potassium channels. So due to closure of these ATP sensitive potassium channels, the potassium ion cannot be escaped from the cell or cannot coming out of the cell. So there will be increase in the potassium ion inside the beta cell, so that there will be increase in the positive charge inside the beta cell, and so due to this there will be the depolarization, and due to this depolarization it activates the calcium channel, and the calcium channel will become open up so that the calcium ion enter inside the beta cell, and once the calcium ion increase inside the beta cell, it triggers the event of release of the granules of the insulin. So that is the mechanism how the insulin is released. Here the calcium channel, that is the voltage gated calcium channels. So that is the mechanism is working behind the release of the insulin, and it is directly related to the glucose concentration inside our body. So here the amino acid having the add-on effect over the insulin release. So here it is the insulin, and this insulin binds to the cell membrane receptors. So this is the cell membrane receptors for the binding of the insulin, and once the insulin bind to the cell membrane receptors, it activates the tyrosine kinase. So once the activity of this insulin receptor substrate or IRS is activated, which leads to the different process and activities inside the target cell. For example, fat synthesis, protein synthesis, growth and gene expression, glucose synthesis, and also there will be development of glucose transporters which are located inside or on the cell membrane, so that the glucose entry will be possible inside the cell at a higher level, or in a more and more number of glucose molecules will enter inside the target tissues. So that is the basic mechanism of the insulin over the target tissues, and how it can lead to the increased utilization of glucose by the target cell. Now the glucose transporters. Here are the different glucose transporters. So mainly the insulin dependent glucose transporter, that is the GLUT4, and it is located in the skeletal muscle, smooth muscle, cardiac muscle and adipocytes, that means inside the adipose tissue. And all other GLUT1, 2 and 3, all three are the insulin independent transporters. The main insulin dependent transporter, it is the GLUT4. Now what is the effect of insulin on the carbohydrate? Rapid uptake, storage and use of glucose, especially by muscle, adipose tissue and liver. So when the insulin concentration is increased inside our body, it leads to increase uptake of glucose. So there will be increase in the uptake of glucose by the muscle cell, adipose tissue and liver. Alright, not needed by most of the neurons in the brain. So insulin is not required for the transport of glucose inside the neuron. In other words, we can also say that the transport of glucose in the neurons is insulin independent. Carbohydrate metabolism effect of insulin on the muscle. Normally during the rest, inside the muscle fats are used. Resting muscle membrane is slightly permeable to the glucose except with insulin. When the insulin is released, now the resting muscle is more permeable to the glucose. When the muscle will use the glucose during exercise, there will be increased permeability to glucose and even without insulin. After meals, there will be increase in the insulin and increase in the glucose uptake. At the resting condition, the muscle will not take the glucose, or in some other word we can also say that the very little amount of the glucose is taken by the muscle at the resting condition. But when we are doing the exercise, the muscle glucose uptake will become increased without insulin. And in the conditions of after meal, when there is increase in the blood glucose level, there will be increase in the blood insulin level, and due to this insulin, the muscle uptake of the glucose is increased even at the resting condition also. So that is what here you have to understand. Here you have to understand that without insulin at resting condition, the muscle will not take the glucose in a much higher quantity, but after meal there will be increase in the glucose level, so that there will also increase in the insulin level, and this insulin will increase the glucose uptake by the muscles at resting conditions also. So that is the different status of the muscle activity or the muscle uptake of the glucose with insulin and without insulin. Not exercising muscle converts the excess glucose to the glycogen and stores it for the further use. Now what is happening at the resting condition? With insulin, the muscle glucose uptake will increase. Now what the muscle will do at the resting condition, that means non-exercising condition of the muscle. Now whatever the excess molecules of the glucose which has been taken up by the muscle, converted to glycogen, and the glucose is stored inside the muscle in the form of glycogen for the further use. That means when there is a need of the energy, then the glycogen will break down. Alright, so that is the carbohydrate metabolism and effect of insulin on the muscle. Now carbohydrate metabolism effect of insulin on liver. So what is the effect of insulin on the liver? It having the hypoglycemic action, that means on liver it is also causing the glucose uptake. So increased glucose uptake which is converted to fatty acid, and this fatty acid deposit into the adipose tissues. It inhibits the gluconeogenesis. That means, what is gluconeogenesis? That means the synthesis of glucose from non-carbohydrate sources. So it inhibits the process of gluconeogenesis. It increases the uptake of glucose, increase glycogenesis, decrease glycogenolysis, and increase storage of glycogen. And there will be decrease in the glycogenolysis, and so that there will be increase in the glycogen storage inside the liver cell. So here it is the diagrammatical representation of the whole of the carbohydrate metabolism which occurs in the liver. So that is the liver cell. Here the glucose molecules which has been uptake by the liver cell in the presence of insulin, and this glucose is converted to the glycogen and stored inside the liver cell in the form of glycogen. And when the insulin is circulating inside the blood, this glycogen breakdown is not takes place. So insulin having the negative effect on the breakdown of the glycogen, so that the glucose will not be released, and the net effect will be the glycogen stored inside the liver cells. Carbohydrate metabolism, effect of insulin on brain and other cells. So brain uses only glucose for the energy. The neurons or the brain tissues uses the glucose without the insulin except ventromedial nuclei of the hypothalamus, and at 20 to 50 milligram percentage blood sugar level the condition is known as hypoglycemia. Now what is the effect of insulin on the adipose tissue? So this insulin stimulates the uptake of glucose and free fatty acid by the adipose cell, and due to this uptake, the glucose and free fatty acid will become converted to glycerol and triglycerides. The net effect of the insulin on adipose tissue is fat synthesis and storage. So once the triglyceride synthesis takes place inside the adipose tissue, it will become stored inside the adipose tissue. So here the insulin having the negative effect on the breakdown of the triglycerides, so that the fatty acid is not released, but the glucagon will promote the breakdown of the triglycerides and fatty acid will become released. We are talking about the insulin. So insulin stops the release of free fatty acid and stops the breakdown of the triglycerides. Effect of insulin on fat metabolism: it promotes the fat synthesis and storage, decrease fat utilization, increase lipogenesis, that means excess glucose which enter into the liver cell is converted to fatty acids. It promotes the storage of fats in the form of triglycerides in the adipose tissues. It decreases the lipolysis and having the anti-ketogenic effect, so it decreases the ketone bodies inside the blood. When the insulin is lack, the glucose metabolism is shifted to the fat metabolism. So here one thing we have to note that when the insulin is present inside our body, the main source of energy it is the glucose, and when insulin is not present inside our body, the main source of energy is become fat. So when the insulin concentration is decreased or no insulin inside our body, it increases the utilization of fats, increase free fatty acid, increase cholesterol, and increase phospholipids. This lack of the insulin also promotes the abnormal activity of the atherosclerosis and it having the ketogenic effect. Okay, so that is what happening when the insulin is not present inside our body, and that is what is happening when the insulin is present inside our body. Effect of insulin on protein metabolism: it promotes protein synthesis and storage, it increases transport of amino acids, it increases translation of RNA and transcription of DNA, so the protein synthesis takes place. It decreases the catabolism of proteins, so the process of protein breakdown has been decreased, and it decreases gluconeogenesis in the liver. Basically it having the protein synthesis type of activity. Insulin lack, that means when insulin is not present inside our body, so there will be increase in the urea secretion, as well as the muscle wasting and weight loss will be the possible outcome when the insulin is not present in the adequate amount or the insulin is completely lack inside our body. So that is the effect of insulin on the protein metabolism. Now the synergistic effect with the growth hormone, both needed for the growth. So for example, that is the weight, and here it is the days. When the growth hormone having only effect on the person, then the weight will become not that much increase, as you are seeing up to 5 to 10 kg will become increase. When the insulin level individually affecting the weight of the person, then the weight is only the same, 5 to 10 kg will become increase. But when the growth hormone and insulin both affecting the growth and development of a person, the weight will become rising around 20 to 30 as well as up to the 50 kilogram. So that is the synergistic effect of insulin with the growth hormone. So when the plasma insulin level increased inside the muscle, what will be the effect? So it increased the glucose uptake and utilization, net glycogen synthesis, net amino acid uptake, and net protein synthesis. Now what is the effect over the liver? It increased the glucose uptake, net glycogen synthesis, net triglyceride synthesis, no ketone synthesis. Now what is the effect on the adipocytes? It increased glucose uptake and utilization, net triglyceride synthesis. So that is the summary. When the plasma insulin level is increased, then what will be the effect on liver, muscle and adipocytes? Now when the plasma insulin level decrease on the muscle, it decrease glucose uptake and utilization, net glycogen catabolism, that means breakdown of the glycogen, net protein catabolism, breakdown of the protein, net amino acid release, fatty acid uptake and utilization. With the presence of insulin, glucose is the main energy source inside the muscle, but without insulin, the fatty acid is the source of the energy. In the liver, glucose released due to net glycogen catabolism and gluconeogenesis takes place inside the liver, as well as ketone synthesis and release. Now in the adipocytes, decreased glucose uptake and utilization, net triglyceride catabolism and release of glycerol and fatty acids. So that is the effect when the insulin level decrease inside our body, and how the different tissues, liver, muscle and adipocytes reacted to this decreased level of insulin. When the plasma glucose level near to the 100 milligram per dl, there will be no any major amount of the insulin is released. After meal, within 10 to 20 minutes, whatever the preformed insulin is released. Once the preformed insulin concentration has been reduced inside the isolates of Langerhans beta cell, there will be new synthesis of the insulin takes place inside the beta cell, and gradually there will be increase in the insulin level, which combinedly both the preformed as well as newly formed insulin is released. Now after the 30 minutes, the newly formed insulin release will become increased. So plasma glucose concentration above 100 milligram per dl increases the insulin secretion, as we just went through this. Which are the factors which increase the insulin secretion? Increase in the blood glucose level, increase in the blood free fatty acid level, increase in the blood amino acid level, GIT hormones, gastrin, cholecystokinin, secretin and gastric inhibitory peptide, glucagon, growth hormone and cortisol, parasympathetic nervous system stimulation, acetylcholine, beta-adrenergic stimulation, insulin resistance, which leads to obesity, and that is also the one of the factor which increases the insulin secretion, and sulfonylurea drugs. Now in which condition there will be decrease in the insulin secretion? So first, when the blood glucose level is decreased, in the fasting condition the insulin secretion is decreased, somatostatin, alpha adrenergic activity and leptin. So all these are the factors which decrease the insulin secretion, and these are the factors which stimulate the insulin secretion. Pancreatic beta cells secrete the insulin when the plasma glucose level increase, so it is the main stimulus for the release of the insulin. Second stimulus for the release of the insulin, it is the increased plasma amino acid which potentiates the effect of glucose. That means the amount of the insulin which is released alone with the help of glucose increase, it is little, but when both the glucose and amino acid having the effect over the pancreatic beta cell, the more amount of the insulin release has been takes place. So GIT hormones, GIP, gastrin, CCK, that means cholecystokinin and secretin, all three or four hormone having the stimulatory effect over the pancreatic beta cells and release the insulin. Glucagon, growth hormone, cortisol, estrogen and progesterone, all these having the stimulatory effect over the pancreatic beta cell. Norepinephrine and epinephrine both having the inhibitory effect over the pancreatic beta cells, so insulin release will not occur, and parasympathetic stimulation stimulates the pancreatic beta cell and insulin release will be the outcome. These are the control of insulin secretion.